All right, welcome to Exam 2 review for Micro. We're going to get started. I just sent out a PowerPoint for the exam review, so everybody should have that. So there it is. Okay. So we'll go through what's going to be on the exam for material. So first and foremost, Enterobacteriaceae, probably one of our biggest weeks of bacteria that we've had. I know that Enterobacteriaceae can get to be tough. There's a lot of them, but what you should know from this group is the TSI and IMVIC reactions for sure. Have to know those. This is something that you guys have already been quizzed on. You guys have had these now a few times in lab, hopefully, so please know those. I've also put up announcements previously on them, but make sure you really get to know those reactions for the different Enterobacteriaceae. That will help you a lot when you get to case study questions and they're giving you all these test results and you're like, oh my gosh, what is this? If you know just some of the key reactions of these, you can pretty much figure out one of the multiple choice answers and get it. So with that being said, also remember, what does it mean to be part of the Enterobacteriaceae group in general? So I don't think, nope, I didn't have this yet written down on here. Let me, I'm going to end this so I can type on here. So again, let's not forget there's 5 characteristics in order to be considered part of the Enterobacteriaceae group. You guys have listed these on the lab report. I know we've had a couple test questions on them, quiz questions. So first and foremost, they are all, of course, gram-negative rods. Second, they are all what we call oxidase negative. Remember, as soon as we know we have a gram-negative rod present, we always oxidase test next. And by knowing if it's oxidase positive or negative will really help determine which way to go. So if you get a gram-neg rod that's oxidase negative, it's most likely going to be one of these Enterobacteriaceae. So then you can start with your IMVIC testing and all that kind of stuff to help figure out which one it is then. Most of these are going to reduce nitrates to nitrites. Most are catalase positive. I know catalase is more of a gram-positive test, but in this case, they are almost all catalase positive as well. And then all of these are glucose fermenters. So those are your 5 characteristics in order to belong in the Enterobacteriaceae group. So don't forget those 5. All right, going back to our slide here. Now, some of these organisms in the Enterobacteriaceae group are listed here: E. coli, Yersinia, Proteus, Salmonella, Shigella, Serratia. There's, of course, many more. There's Citrobacter, Edwardsiella, Morganella. There's a bunch more. These are the main ones that we've been kind of focusing on and that I want you to still focus on for the exam. So I'm just going to list off some broad characteristics of each one, but be sure, again, you go back to your PowerPoint, your, lab stuff, your book, things like that, anything like that to kind of know more details. Study guides that you've completed are a great resource as well. So in general, E. coli tends to be beta hemolytic, not every single one of them, but most of them. And they tend to be lactose fermenters on MacConkey agar, meaning they'll have that kind of purple colony on MacConkey. If they were to grow on EMB agar, they would have a green metallic sheen on EMB agar. So that's kind of the general gist of what E. coli looks like on agar. Remember, E. coli is hugely known for being indole positive. That's a really big test result with E. coli, is that indole positivity. And in fact, overall, its IMVIC reaction is ++, neg-neg. So it's positive for both indole methyl red, negative for Vogt-Proskauer and citrate. So there's kind of the indole. TSI reactions, it can have a couple different ones. A over A, I think it can even have K over A. So just kind of look up the TSI reactions there as well. So that's just general. Now, for E. coli, there was lots of strains of different E. coli. We have E. coli as normal flora in us, in our intestines. But if you were to pick up a different strain of it, then you can become ill. Mainly, it's going to cause a lot of diarrhea type stuff. But E. coli is very much known as being the #1 cause of UTIs. So, but we can also see it with wound infections, septicemias. There's other things where E. coli will pop around, but don't forget to look up the strains of E. coli, like enterotoxigenic, enterohemorrhagic, enteroinvasive, enteropathogenic. So we learn those different strains, so make sure you go back and just keep a look at that. All right, for Yersinia, Yersinia is typically, it can be non-modal at times. Most Enterobacteriaceae are modal. Yersinia is one of the ones that can be somewhat non-modal, just depending on the temperature that's occurring. We have learned that stin agar is used to help identify Yersinia and have bullseye colonies on there. Now, there are two main Yersinia ones. We have Yersinia pestis, which of course is passed by fleas from infectant rodents, and it causes the plague. And then you have Yersinia enterocolitica, which causes enterocolitis, what it's called, which is a nasty diarrhea. So those are your two main Yersinias. They're also said to have a safety pin appearance on gram stain because it just doesn't take up the stain evenly, so it can sometimes look like a safety pin look. So, all right, Proteus. There are two Proteuses in general, but both have this really unique swarming ability on agar. So I think hopefully you guys have seen that and smelled Proteus. Proteus smells so nasty. You can always smell when Proteus is growing. So lots of swarming ability on agar. Typically, it's a non-lactose fermenter on McConkey agar. Proteus is very much modal because of the swarming, hugely modal because that's how it swarms across the plate. Let's see. As far as the species, we have Mirabilis, which is indole negative, and you have Proteus vulgaris, which is indole positive. And you guys have answered that question a few times. So the indole results help separate them. Remember, though, Proteus has lots of other chemical reactions. It is hugely urease positive, and then you can know the IMVIC and TSI reactions. Proteus is also known to produce H2S at times, so H2S positive. And then as far as diseases go, it's lots of UTI stuff, diarrhea, like some other things. It can pop up here and there, but I'd say UTIs are a big one we see it with. And then wound infections, Proteus could be wound infections too, so some stuff like that. Salmonella is a non-lactose fermenter. It is H2S, very much H2S positive, so sometimes it has that black center. If you look at HE agar, hectone enteric agar, it'll have a blue-green colony with the black center to kind of screen for it. We all know salmonella is a huge cause of food poisoning, and there's lots of different types of salmonella. So you just kind of have to look into that, and then I think that's it as far as what I want to say there. Shigella, also non-lactose fermenter. This one is very much non-modal, doesn't move, whereas the rest tend to be movable. Again, on HE, it'll be blue-green, just no black center because it's not an H2S producer. So it's typically like K over A, TSI kind of thing. So look up the IMVIC reactions there as well. Again, Shigella, another big food poisoning cause, if I didn't say that already. Maybe I did. Serratia is a late lactose fermenter, and sometimes as a result of that, it can have a red pigment on McConkey agar. Otherwise, Serratia is just kind of, sometimes we see in UTIs, sometimes nosocomial stuff. We don't see it with a ton of things, but you know, it is what it is. So I think that's all you have to know there for Serratia. There was another one, Klebsiella. I'm surprised I didn't have Klebsiella listed. Klebsiella, very much a mucoid lactose fermenter. So that's the snot on the plate one I always like to talk about. Now, there are two different main ones. There's Klebsiella pneumoniae, which of course can cause pneumonia, and along with some other stuff. This one is indole negative, and then if it's Klebsiella oxytoca, it would be indole positive, which is the other main Klebsiella. Again, know your TSI and IMVIC reactions for these. So that's kind of just a big, broad overview of some key features of each one. The important thing is getting to know your test results. There's those. I put up a chart in the announcements. Make sure you look at that and the different test results. So that will help separate. Okay. Going on, other gram-negative rods that we've been learning, Campylobacter, Helicobacter, these are separated out because these are oxidase positive. So they're different in that they are oxidase positive. Both are said to be kind of curved. In fact, Campylobacter is nicknamed seagull wings because of the curved gram-negative rod look it has under the microscope. This is the one that likes a little bit extra warmth incubator, so 42 degrees. Campylobacter jejuni, #1 cause of gastroenteritis in the U.S., bacterial gastroenteritis, I should say. So it definitely comes from food poisoning, especially with like chicken. There are other Campylobacters here listed that more are linked with periodontal disease. And then Helicobacter is a gram-negative rod, very strongly urea positive and well known at causing gastric ulcers. So that's where you get Helicobacter. We learned anaerobes. Anaerobes, there are some big groups here. We have Clostridium, which are all gram-positive rod spore-forming anaerobes. You have 4 different ones. Make sure you still know the diseases that go with each one and anything else here listed. I kind of try to list the unique features. So Clostridium tetani is, of course, tetanus, which is lockjaw. This one is known as their spores of being terminal, meaning at the ends of the rods. Clostridium difficile, what we always say C. diff. Oh my gosh, you'll hear of this so often in the hospital world. Very much linked with causing severe diarrhea, especially after a round of antibiotic therapy, which kind of wipes the normal flora clean, and then this flares up and causes diarrhea. C. diff is very much passed in hospitals and nursing homes, so usually patients are in at least contact precautions so that we're trying to minimize who we're passing it to. You can do a quick kit test if we think it's C. diff, looking for the toxins that it produces. Otherwise, if you're going to grow it out, there's a special agar used for it called CCFA agar, and they will have yellow colonies on that agar. Clostridium perfringens is causing gas gangrene. This is unique in that it has a double zone of beta hemolysis on the agar plate. And then finally, Clostridium botulinum, which causes botulism, food poisoning. So that's the whole Botox deal. Oops, didn't mean to move there. All right. Another group was the Bacteroides fragilis. Well, it's not a group, I guess, but Bacteroides in general is gram-negative rod anaerobes, lots of infections. Typically, this Bacteroides fragilis is like your most commonly seen anaerobic infection. So if it has infections, it's typically below the diaphragm, very foul odor. There are some unique agars here to help grow it because it loves to grow in the presence of bile. So BBE, LKB both have bile in them, so it will grow. And then again, as we learned in the discussion from week 5, very resistant to those 3 antibiotic discs, and that can help kind of use to identify it. Fusobacterium nucleatum, another gram-negative rod, but this is very unique and it has tapered ends. That's kind of some key words there. Typically head and neck type infections, so kind of above the diaphragm. Prevotella and Porphyromonas, both gram-negative rods, both will have brick red fluorescence, both are head and neck type infections. And then Propionibacterium is a gram-positive rod. This is the one that's called Propionibacterium acnes because it is known to cause inflammatory cystic acne. It also can be somewhat normal flora of the skin. As a result, it can be a blood culture contaminant because it can be picked up off that skin. And week 6 was about mycobacteria. So mycobacteria is a unique group of bacteria in that they don't stain, and that's because their cell walls are different. Instead of having the nice peptidoglycan layer and all that in their cell walls, they have mycolic acids. And so they will stain really, 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 really weakly, like gram-positive maybe, but otherwise they're just not going to pick up the gram stain. So instead, we're going to acid fast stain them. And so again, there's Ziehl-Neelsen and Kenyon are the 2 acid fast staining methods. You still need to remember those. Ziehl-Neelsen is known as the hot method, Kenyon as the cold. Or there's a fluorescent technique called auramine-rhodamine, which will go and bind to the mycolic acids and then fluoresce. So these are the ways in which to stain and identify mycobacteria. As far as selective agars to grow mycobacteria on, we have Middlebrook and Lowenstein-Jennings. And some of the big mycobacteria that we learned was the Mycobacterium tuberculosis complex. So these are the ones that can cause tuberculosis. So we have the main guy, which is tuberculosis. Then you have Mycobacterium bovis and Mycobacterium africanum. Another big group complex that was in here was Mycobacterium avium complex. So there was a bunch in there. These are specifically kind of targeting immunosuppressed people, like AIDS patients are notorious for having this picked up with them, with their immune system being so low. And then the last one was Mycobacterium leprae, causing leprosy, otherwise known as Hansen's disease. And then we categorize our mycobacteria, as we know, 4 groups, all depending on how slow they grow and if they develop pigment or not. So again, you need to know these. Group 1, photochromogens, slow growers that develop pigment following light. That's why it's called photo, for light. Group 2 is scotochromogens, slow growers that develop pigment in dark or light. Group 3, non-photo, meaning non-pigmented, if you think of it that way, and slow growers. And then finally, group growers are only rapid growers that we really have. Now, there were a lot of other mycobacteria. Do go back to your original PowerPoint, link the ones with each group, and just know brief bits on what they are seen in for as far as infections go. Like, I'm thinking right off the top of my head, you should be knowing Mycobacterium marinum, Mycobacteria kanzensii, Mycobacteria zinai, that's the one with the X, E, Mycobacterium scophaceum. Those are some of the bigger ones besides the ones that we just mentioned right here. So just go back, quick look. I don't want you to be experts. I don't want you to memorize everything. Just memorize, like, if it's a respiratory cutaneous disease and then maybe what group they fit into. That should be good enough. All right, so that's it. Just a nice big overview. It kind of gets you to go back to the original content and restudy. I hope you guys all have a good week. Otherwise, if you have questions, please, again, as always, let me know. But good luck on your test. Thanks.